All right, let's go ahead and talk about clinical treatments for surgery, different types of things that must be considered when a patient presents with cancer. How do oncologists, clinicians make decisions about how to approach treatment? And also, this will lead into a discussion about different strategies for coming up with new therapies for dealing with cancer. So one of the big questions faced by a clinician who's presented by a patient in the clinic simply is whether they should treat the tumor or the cancer. And that might seem kind of like obvious, right? You might say like, well, of course, if somebody has cancer, you need to treat by doing something about this tumor. But I think as you've learned to appreciate over the course of the semester, especially as we've gotten into the last few weeks where we've talked about the physiology of how tumors behave in the body, you realize that oftentimes it's not a very straight ahead type of decision. So clinicians often have to think about tumors in three categories. One are what we call indolent tumors. So these are tumors that based upon different types of biological markers, or maybe the location of the tumor, it appears that this tumor has very low invasive or metastatic potential. And so therefore, it could very well be that this tumor, this benign tumor, may remain benign for the rest of the patient's life. Right? So this would be the case, especially if you're dealing with, let's say, an elderly person who presents with a benign tumor. You may feel that any kinds of therapies or ways of trying to treat the tumor might actually have more harmful side effects than just allowing the tumor to stay put. Again, if the biopsy and the pathology comes back from the lab and says, oh, there's very little evidence that this tumor is getting ready to metastasize. You might also consider that if it's a tumor that's actually found in a younger person, right, and it turns out that it doesn't look like it may be very aggressive, maybe the physicians would just prefer to kind of do this watchful waiting approach and just see over a few months what's actually happening with this tumor. Again, because it might be that surgery, depending upon where this tumor is, might have greater risk of causing life-altering types of complications. So somebody who's in their early 30s, right, you don't want them to undergo surgery and suffer for the rest of their life because of things like damage to nerves or other tissues. The second category might be highly aggressive tumors. So these tumors, by the time that the patient presents, they have already metastasized. So in that particular case, the oncologist might feel that dealing with the original primary tumor is a secondary issue compared to how to deal with the metastases. Remember, we learned that the majority of people who succumb and die from cancer die from secondary metastatic lesions in their body, not normally the primary tumor. And then the third category, these might be intermediate grade tumors. So by the time the patient has presented, the tumor has already begun to grow and develop. Maybe there's some evidence that it could be heading towards being more aggressive, but maybe it's still at that point where the patient can try undergoing different types of chemotherapies, right, using cytotoxic agents to try to biochemically kill the tumor preferentially compared to the normal tissues in their body. So these are different considerations. So let's investigate some of these. So I should point out that of the three different sort of categories of the tumors that are presented, number three, the intermediate grade tumors, because of their nature, meaning that they haven't metastasized yet, so there may be a good possibility of trying to treat the patient with chemotherapy. This number 3 category then presents the greatest choices for different types of treatment methods. And so the key here then is to really understand the specific biology of the tumor. Right? Don't treat it as though it's just, oh, it's a mass that needs to be dealt with. Right? If you want to have some ability to have different options, if you understand the biology of where this tumor came from, right, that can open up many other treatment options. So this is some data looking at historically how the advent of what we consider now some traditional therapies for cancers have impacted mortality rates of different types of cancers. I don't think any of you would be surprised when I say that development of different types of therapies and things like this have been more successful for some types of cancers compared to others. So on the left is an example of mortality data, looking at a span starting from the 1930s all the way to 2006, following these 4 types of cancers, which are good examples of cancers where over time, right, as we've learned more about cancer, understand how to treat cancer, death rates of these cancers have gone down. And so they include things like stomach cancer in both men and women, uterine cancer in women, and colon and rectal cancers in women. Okay. A lot of this has to do, again, with better diagnostic techniques. Being able to identify cancers earlier can lead to more intervention at an earlier stage. Many of these also have to do with just lifestyle changes, better understandings of things that people should or should not do in their daily lives. And things like, for example, stomach cancer rates dropping because of things like more access to refrigerated foods, better refrigeration, less dependence on storage of foods where they might go bad, moldy, and result in different types of naturally occurring fungal toxins like aflatoxins that are linked to stomach infections and inflammation and cancer. So we talked about H. pylori as a very good example, that particular pathogen that's associated with food that's not stored well, right? That can cause stomach cancer. But if you look during that same span, 1930 to 2006, there are several examples of cancers where rather than the mortality rates going down, mortality rates have somewhat plateaued or even increased dramatically over time. So cases like colorectal cancers in men, right, they went up in the death rates went up in the 1940s and have pretty much stayed stable. Maybe they're beginning to come down in the 1990s and beyond. But things like lung cancer, bronchial cancer in both men and women have really skyrocketed, right? So remember, these lines represent mortality rates. So this is actually a measure of how many people are dying every year from these. So this is a really amazing set of data because it really highlights how the introduction or the popularity of cigarette smoking has taken its toll. So before the 1940s, cigarette smoking, even though it was popular, it was not as commonplace in women compared to men. But during World War II, when the US government was basically trying to get people to join the war effort and join manufacturing of things like ships and planes, missiles, whatever. And then you actually had the soldiers being sent overseas to fight in World War II. They were given, their rations that oftentimes contained government provided cigarettes. Right? And even the people who stayed at home back in the States working in the factories were given cigarettes, right, to keep them productive. And so as a result, the popularity of cigarette smoking really shot up in the 40s. and the 50s. And what you see, remember, that just because you start smoking a lot of cigarettes doesn't mean you will actually develop cancer right away. We've talked a lot about this idea that it takes many years for a cancerous tumor to form. And that's why you see the lag here, where you have it peaking. The death rates in men from cigarette smoking peaked in the mid 80s, right? So about 40 years after World War II. So if you imagine men started smoking a ton in the 40s, and let's say it took 20 years or so for lethal tumors to form, right? And then maybe these men would undergo certain treatments, things like that, and they would eventually succumb 10 to 20 years later after their tumors were first diagnosed. And same thing with women. That's the pink line here. So the way that traditionally we've dealt with cancer is through different strategies that can be categorized as either surgical, right? That's where, very obviously there's this observation that if you can identify a small tumor and remove that small tumor from the body, then the resection of that primary tumor in time helps prevent metastases. Number two is radiation. So people have undergone radiation treatment for a very long time. And so oftentimes following surgery, in order to try to kill the remaining residual tumor cells in the body, radiation is often used. And so when you combine one type of treatment with another type of treatment to get the benefit of them working together, we call that an adjuvant therapy. And radiation as an adjuvant therapy following surgery has proven to be tremendously successful. And then the third strategy is chemotherapy. And it should be pointed out that historically, many of the most popular chemotherapeutic agents that are used to treat cancer were discovered kind of blindly by luck from identifying them in natural sources that were then synthesized in the lab, or perhaps they were synthetic molecules that then by chance, through some testing, were found to actually have good anti-tumor properties. So historically then, the development of new chemotherapeutic agents, drugs, things like that, have happened without much real a priori understanding of mechanisms of how they work. A priori means many of these drugs were developed not necessarily in a rational way, where the researchers knew what they were going after, but oftentimes they were stumbled upon and then improved upon. Okay? And only after they began to be used as drugs did a better understanding of mechanism actually come about. So here are some examples of newer kind of developments in surgery as well as radiation. Okay, so those are the first two kind of strategies we're talking about. These days, it's very common that patients who undergo surgery for removal of tumors, especially if the tumors are buried very deep inside the body in places where traditional hand surgery is dangerous. Right now we have robot-assisted types of surgery. So this is a Da Vinci robot where you can see on the left, right, that here's the dummy representing the patient on the table. And in this case, there are no actual human being surgeons surrounding the patient. You just have the robot. And then the robot has these multiple arms, and these arms have very fine little manipulating attachments at the end, where they can do very fine, very delicate surgery. And they are controlled, you can see here, by a little control station. So here's the operator working the control station, right, where they can actually have a good three-dimensional stereoscopic view that's highly magnified of the tissues inside the patient. Right, so this is actually kind of funny. When I was a graduate student at University of Southern California, that's when these types of Da Vinci robots came out, and they actually did a demo at our cancer center, and they invited everybody to come and see. And they offered free pizza. That's how they got all the grad students to go. So I went for the pizza. But anyhow, they had this really neat demo where on the table they had a little pile of Fruit Loops, right, like the cereal, and they showed how delicately the robot could manipulate these little Fruit Loops, right, controlled by the person, the skilled surgeon operating the console there. So that was almost 20 years ago. And so I think these robots have gotten even more sophisticated, right? So this is a big advancement in surgical techniques. Also with radiation. Okay, so traditionally, it's been known for a long time that if you hit tissues with high doses of radiation, not surprisingly, there's tissue damage. And as we learned, if the damage to the genomes of the cells cannot be fixed, then the normal healthy body cells should undergo apoptosis. So in cancer cells that are rapidly dividing, remember, the cells, especially aggressive tumor cells, are all about dividing, right? They've stumbled upon mutations that are just accelerating the rate at which they go through the cell cycle to get from one mitosis to the next mitosis. And part of that is they've disabled many of the checkpoints along the way that would normally prevent the cell cycle from going around and around and around. So we've discovered that if you treat these cancer cells with high doses of radiation, they tend to be very sensitive to the radiation, even more than your typical normal healthy cells. They undergo what we call mitotic catastrophe. Right? That sounds very dramatic, but it's very appropriate. What it means is as the cells are plunging forward through mitosis, right, they get to a point where the mitosis just isn't happening anymore properly. It's a disaster, right? It's a catastrophe. And these rapidly dividing cancer cells just simply can't do it anymore. And they fragment and they die. Right? So why would you think that tumor cells would be more sensitive to this type of high-dose radiation damage, inducing genomic instability? And so it turns out it's because, remember, a normal cell, if it gets damage from radiation, it has different checkpoints, different tumor suppressor genes like P53 that would halt the cell cycle and try to allow that cell some time to fix the problem. But an aggressive tumor cell that has lost all of those breaks, it won't bother spending time trying to fix the problems, right? It's just going to keep plunging forward. And so if those problems are too great, they might be problems introduced by radiation that actually are damage to an extent that even the tumor cell cannot deal with and successfully undergo mitosis. So that's why tumor cells are more sensitive to radiation than normal cells in our body. Okay. So it used to be that you would just beam the body with high doses of radiation, right, to try to get to the tumor. So of course that's bad, because even though the normal healthy cells might still have their tumor suppressor pathways in them, right, this is still not good to be exposing the majority of the tissue in the body to high levels of radiation, because then it's no different than when you're getting sunburned from, let's say, being out in the sun too long, right? And every time you get sunburned, you're going to have some cells that don't undergo apoptosis properly and might hang around. Right? So one of the problems with radiation has always been off-target effects, actually the possibility of causing more tissue damage in the attempt to try to get rid of the tumor. So recent advances have to do with different ways of focusing the radiation on just the tissue that you want to target. So this involves what we now call a focused beam radiation. So that's when you actually, instead of a big dose of radiation, it's actually focused in beams that maybe each beam itself is a low enough dose of radiation that any tissues that they go through would not be damaged too much. But if you're able to focus the two beams together and have them cross right at a particular location inside the body, right, then at that crossing point, the dose of radiation is high enough to cause damage. Right? So obviously that means you try to focus the two beams so that they focus on the tumor wherever it is inside the tissue. Another way of doing it is what we call brachytherapy or seed radiation therapy. So this is when, as you can see, they have little pellets of highly radioactive material. So these are oftentimes different radioactive metals. Right? And again, each little seed, right, can then be inserted physically inside the body. So here is an X-ray of a prostate cancer patient who is undergoing this type of brachytherapy. You can see each one of these little white dots is one of these radioactive little pellets. And each one of these little pellets, the amount of radiation that they emit is not strong enough to cause a lot of damage. But if you carefully insert a whole cluster of these little seeds right into the tumor, for example, then the combined amount of radiation, right, which is highly localized, can target the tumor cells and hopefully induce mitotic catastrophe. All right, so let's go back to this slide then. So that's just some advances in surgery and radiation, but you can probably imagine that, even though the robot might seem very impressive, or using focus beam radiation might seem very impressive, right? Really, you're talking about still very sort of basic concepts, right? Of physically removing bad tissue that you don't want, or using radiation to kill that tissue. So therefore, surgery and radiation, there have been many improvements in methodology, but neither of these are really concerned with understanding where the tumor came from in the first place. Right? These approaches just deal with getting rid of the tumor. So the third strategy then, chemotherapy, as I mentioned before, you have the most room for making different types of advancements in being able to target phenotype-specific cancers. Right? That means by understanding biology of the different tissues that gave rise to these tumors and how these different specific types of tumors behave differently, then you have a real chance for developing novel drugs, novel compounds, for example, that can interfere in very pathway-specific mechanisms. So this is very exciting because this gives you the most opportunity then for really going after specific cancer subtypes. So here's some examples then of different compounds. And these are compounds that have been used for some time now, but just to give you an introduction to the ways that different types of chemotherapeutics work. So retinoic acid is a very classic cancer treatment. Retinoic acid actually induces cells to undergo differentiation. So it's not that it causes the cells to die or apoptose, it just stimulates undifferentiated cells to go ahead and actually commit to differentiating. So here on the left, you have some example of some promyelocytes. So these are immature white blood cells. And so they typically have this very large nucleus in them. This is what they would look like in the bone marrow, or maybe when they're just released from the bone marrow and they're still, need to be differentiated into a final type of cell. So not surprisingly, when you have different types of cancers involving your immune cells, like B cell, leukemias, lymphomas, those types of things, what you see are the cancer cells look like this, right? They are undifferentiated and they're just preoccupied with proliferating like crazy. So retinoic acid then, if you treat these promyelocytes with this RA, it stimulates them to actually go ahead and finish differentiating. So here they've become neutrophils. And so you might think, well, that's kind of weird, because why does it matter if you're trying to just force cancer cells to differentiate, right? Why might this be a good anti-tumor strategy? Okay. And this has to do with this concept that we learned before, that at any given time, a given cell is either focused on doing its differentiated job, right, and becoming the fully differentiated cell it's meant to be, or it's focused on proliferating. So if you have a cancer cell that's very focused on proliferating, if you can encourage it instead to give up with the proliferating and try to be more differentiated, right? That's one way that you can try to put a halt to the tumor cells dividing. So here are some small drugs that target DNA. Not surprisingly, many of the compounds that are used as chemotherapeutic agents have DNA as their target. So some of these act by forming adducts or cross-linking DNA. So these might involve alkylating agents, such as these pictured here. So in this case, they might form cross-links. In other words, they'd attach to the DNA bases and maybe not attach just at one place, but also attach at another place, causing strands of DNA to get sort of cross-linked or stuck together. This drug down here, carboplatin, is a very common first-line chemotherapy that's used for many types of cancers, especially things like breast cancer and ovarian cancer. Over here, these are drugs that work by mimicking or resembling nucleotides. So you can see here, especially these two like gemcitabine, they look like nucleotides, right? They look like DNTPs. They have an amine base, a sugar ring, and in this case, even a phosphate ring. So these, you can imagine, can incorporate into DNA that's being replicated. So as the DNA is being replicated and the template strands come apart, and you have your DNA polymerase beginning to read the template strand, trying to make a copy, it's going to be pulling all these nucleotides that are floating around to add the next A, T, C, or G where it belongs. So if you have this stuff floating around, then these will be incorporated. But once they're incorporated, that really messes up the DNA. So again, these are small compounds, right? They're not terribly complicated in structure, so they have good properties. Because they are small, they can easily get into the nucleus of the cell. They can easily incorporate into the DNA, right? But in these cases, when they incorporate into the DNA, it messes up the DNA structure. Right? And the fact that they work by incorporating into DNA, right, that should tell you something about what types of cells would be more sensitive to them. A normal healthy cell whose DNA is tightly wrapped up around nucleosomes, right, these drugs wouldn't have the ability to get in there and incorporate as easily compared to, let's say, a rapidly dividing cancer cell who's always undergoing mitosis and always copying its DNA. Right? That would be the opportunity for compounds like this to integrate. Let's take a look at this carboplatin a little bit more. So carboplatin is based around platinum, right? So in this case, it was discovered in the mid-1800s that there was this compound. They just called it Peyron salt. For many, many years after that, there was no real clinical use for this compound. But in the mid-60s, some researchers made a very lucky observation that had nothing to do with them wanting to discover new cancer compounds. So they realized that when they were running gel electrophoresis, so they're running gels that had electrodes in them in a buffer, what they observed was that oftentimes if their gel chamber wasn't very clean, there might be some bacteria that was growing inside of the gel chamber. But yet right around the electrodes, the platinum electrodes, right, the plus minus cathode and anode, they discovered that the bacteria would not grow, right? And it's not because of the electricity, because the electricity is flowing through the whole thing. There's something going on around these electrodes. And what they realized was because of all the electricity, you would actually have the platinum from the electrodes kind of wearing off and forming this compound cisplatin. And so they published this paper in Nature. You can see where the paper is just about this interesting observation that E. coli is inhibited from growing when you expose them to these various platinum products coming from the electrode of one of these gel chambers. right? So it's kind of neat because you think about it, when this was published, people probably thought like, oh, that's interesting, right? But who would actually realize that the reason why these E. coli were dying was because the cisplatin was integrating into the bacterial genomes, messing them up, right? And so cisplatin does that. So this is a more accurate three-dimensional space-filling model of the cisplatin, right? Another variation is called carboplatin. And so cisplatin and carboplatin both are very, very popularly used drugs for treating tumors. You can see whether you're talking about cisplatin as pictured here or carboplatin, the way they work is they form adducts in the DNA. So they've joined this one molecule of cisplatin has joined here, right? And because it's linked these two neighboring nucleotides together, right, it distorts the natural alpha helical bend of the DNA. So of course, the cell will try to fix this. It will have enzymes that come and try to snip out this defective piece of the chromosome so that you can fill it in and replace it again. But if you imagine a high dose of this cisplatin, meaning not just at this one spot in the DNA, right, but all over the DNA on every chromosome, being all kind of messed up like this, you can imagine then the cell can't deal with that. It cannot repair it. And again, the cisplatin would hit cells that are rapidly dividing the hardest, because those would be the cells where the DNA is opened up and accessible for the drug. So a lower dose of drug would have a higher effect in those tumor tissues. And that's very important because you can imagine the normal healthy cells of the body, it's not as though they're somehow resistant to the cisplatin, right? So if you have too high a dose of cisplatin, you are actually killing off many of your healthy cells as well. And that's a very common theme when dealing with chemotherapy therapies, right? It's almost like a game of chicken. You're trying to give the body a high enough dose of these chemotherapeutic agents so that the tumor cells are actually getting hit really hard, right? But all the normal tissues are also getting hit pretty hard. So the idea is you got to find that dose that's high enough to get the tumor to shrink quickly, and then when it reaches a certain point where it's shrunk away, you quickly take the patient off the drugs before the drugs actually kill the patient, because all the normal cells in their body are also being affected. So as I said, cisplatin and carboplatin, they have similar what we call efficacies. Efficacy is just a reference of how well a drug works. But carboplatin tends to be better tolerated by patients, meaning side effects are not as great. When we talk about side effects, oftentimes we are talking about things that you associate with patients undergoing chemotherapy. Remember, the cells in their body that are the ones that are rapidly dividing are the ones that get hit the hardest oftentimes by these types of DNA targeting drugs, right? So that's why things like the hair follicles, right? The hair follicles that are constantly dividing to grow your hair, they get hit very hard. And that's why in patients, their hair falls out. Or the epithelial lining of the GI tract, right? Which we learned is constantly being replenished by stem cells dividing and giving rise to epithelial cells, right? They get hit very hard. That's why the patient's GI tract gets very messed up and they experience things like nausea, loss of appetite, weight loss, things like that. Other small compounds don't necessarily target DNA directly, but they target proteins in the cell. So here's a couple examples. This is an etoposide, which is a topoisomerase inhibitor. Topoisomerase is the enzyme complex that unwinds the DNA during S phase, during DNA replication, right? Because remember, the alpha helix needs to be unwound, so you have the two strands. So topoisomerase is the enzyme complex that does that. So this etoposide drug stops that enzyme from working. So you stop the cell from being able to copy its DNA. This over here, this bortezomib, is a proteasome inhibitor. Remember, inside your cell, you have the proteasome complex, which is constantly in charge of recycling proteins to basically break down used or spent proteins or damaged proteins and turn them back into amino acids that then are the resource to make new proteins, right? So if you stop the proteasome, the recycling process from happening, the cell runs out of raw materials to make new proteins. These two compounds on the bottom target our microtubules. And so you can see these two taxanes like paclitaxel and vincristine, right? These are very complicated compounds, right? And you can imagine, how did anybody in the lab come up with these crazy structures? And it turns out that they don't come from man-made structures. They were identified as natural products. Right? So this is a great example, sort of a segue into appreciating then how many of these important compounds come from nature. So these are drugs, the taxane and the vincristine. They both target the microtubule proteins. So remember, during mitosis, the chromatids are lined up along the metaphase plate, and during anaphase, they're linked up to the microtubule spindle fibers, the big green fibers over here, that then pull apart the sister chromatids, so you end up with two daughter cell nuclei forming, right? So if you can interfere then with these microtubules, the spindle fiber working properly, yeah, this is a great way to prevent mitosis. So this is just a reminder. The microtubules are made of these alpha and beta tubulin monomers. So on the side view of a strand, it would look like this. If you look straight down in it, it's this hollow cylinder, so you can see here. Okay. So remember, the microtubules have a polarity. They have a plus end, a minus end. And as the microtubules grow, they're growing because more of these alpha and beta tubulin monomers are added to one end. Right? And when it's time for the microtubule to shrink, for example, when it's pulling away the sister chromatids, right, it's shrinking from the other end. The polymer of the microtubule is broken down so that then the microtubule is shrinking and it's pulling on the sister chromatid off to one side. So taxanes bind to the beta tubulin monomer. Drugs like vincristine bind to the alpha tubulin. Okay. And so this is how they work. So paclitaxel, again, here's this amazing compound structure. You know, if you have flashbacks to taking organic chemistry and exams where you had to derive structures, this would be like a nightmare, right? If you imagine, like, you got a blank sheet of paper, on the upper left you have like a few carbons, and on the lower right you have this thing, and then you have to somehow synthesize this in like 12 steps or something, right? But Mother Nature, over the course of evolution, comes up with this stuff, right? So this drug was isolated from the bark of this tree, the Pacific yew. So the name comes from the genus of this tree, that's Taxus. Right? So it was discovered in the early 1970s. Back then, the NIH had a lot of money and a lot of willingness to just throw money at sort of these big fishing expeditions. And so they had this big initiative where they said, we have millions and millions of dollars. You could apply for these grants to just go out and take samples of plant material and try to screen any of these compounds to see if they have anti-cancer properties, right? What an amazing time that was. These days, you would never have those types of funding opportunities, unfortunately. Right? But this gave rise to the discovery of this paclitaxel from the bark of this tree. And it was shown in 1978 that this compound has anti-leukemia effects. But the problem was, in order to make purified paclitaxel, they needed something like 60,000 pounds of bark from the tree in order to purify enough of this drug to actually meet the drug demands. Right? And so that made the Pacific yew tree, that put it on the endangered species list, because they were harvesting this tree for the bark. Right? So then there was a need for some lab somewhere to figure out how to synthesize this in the lab, right? So you don't actually have to take bark and extract this compound. And Bristol Myers Squibb won the contract for the synthetic production of this paclitaxel. And so the market name they gave it was Taxol, right? And that was in the early 90s. And so for the last few decades then, Taxol has been one of the most important drug chemotherapy agents for fighting cancer. So I think it's wonderful to think that it all came from just the screening project of looking for natural compounds. Right? Remember, I mentioned this to you before. When you think about drugs and medications, the average person doesn't think about, like, well, what does it really represent? Right? Because remember, all of these small compounds, or not so small, if you look at this paclitaxel, it's still pretty big, it's very bulky, represent complicated three-dimensional shapes. Right? And in this case, it's a complicated three-dimensional shaped molecule that does not exist in your body normally. And your body does not know how to make this. But when it's introduced in the body, it can fit somewhere against some protein, right? And essentially it acts as a poison because it's interfering with a normal cellular function, right? The management of the microtubules. So if you think about that, all these types of little structures, there's so many of them still out in nature, just waiting to be found. So in this case, the Taxol drug, it binds to the beta tubulin subunits, the monomers, and prevents depolymerization of the spindle fibers. So the spindle fibers cannot retract. And so as a result, anaphase cannot happen, right? Because the chromatids cannot be pulled apart. So if you think though that, wow, that Bristol Myers Squibb company, they were really lucky getting the patent on this Taxol, that's probably the end of story with the Taxol, right? Not quite. Because even though that Taxol looks like a big bulky compound, right, compared to many large macromolecules in the body, it's still considered a pretty small compound. Right? And so when you give this to a patient, it floats around in the blood, and you're hoping that enough of it will actually end up inside the cancer tumor, right, as it's floating around. But just as with a multitude of other small compounds that are floating around in the blood, right, paclitaxel is cleared very quickly by the liver, right? The liver and the kidneys. So the kidneys especially are constantly filtering the blood, and any small little compounds, right, get filtered out and are then expelled as part of urine. So that means that some of these chemotherapy drugs, you actually have to give the patient a much higher dose than ideal, just to make sure that after they've essentially peed out or metabolized most of the compound, enough of it still is actually reaching the target. So that's true of Taxol. And so, as I already mentioned, it's not surprising that Taxol is highly toxic. So in the start of the early 2000s, a company called Celgene, right, made the discovery that if you actually took this paclitaxel and already bound it up with albumin. So albumin is the main protein in our blood serum. So albumin, it's something like, of all the proteins in our blood, albumin makes up like 70% of our blood proteins. And the function of albumin is actually to carry around small compounds so that they're not quickly eliminated. So in our natural environment of our body, these would include things like vitamins or other types of signaling molecules. They bind to the albumin because albumin has several different pockets on its three-dimensional structure that regularly bind small compounds and carries them around so that they stay longer in your blood. I can't remember if I mentioned this to you, but something like, for example, when you take ibuprofen, right? Ibuprofen is cleared very quickly by your blood, right? So a lot of times you take a dose of something like Advil, right? And within something like, an hour or so, you've lost 70% of it in your urine. So for drugs like paclitaxel, if you go ahead and just stick them onto albumin, you get an interesting situation. So now you can get away with a lower dose of this toxic drug because more of it stays in the system being carried around by the albumin. And so eventually, right, they do fall off the albumin. And so that way, that's how you actually enable them to get into the target tissues. So you might think, well, you know, if Bristol Myers Squibb owns the patent on the Taxol or the paclitaxel, then maybe, you know, there's only so much you could do with this idea. But the interesting thing about intellectual property, especially when it comes to things like drug development, is if you can actually take something like this paclitaxel and attach it to another molecule, in this case albumin, then your combined molecule, right, is now something considered to be new because it's a new chemical composition. And so therefore, you can patent this as a new drug. And so that's what they did here. So this combined albumin paclitaxel as a drug is given the name Abraxane. And so it was approved for use in 2005. And now sales of it in 2007 exceeded over like $900 million, right? So it's really interesting to think these are interesting ways of thinking about how you can take existing compounds and push them forward for new drug development. So the other drug we looked at was this vincristine and vinblastine. So vincristine, again, very complicated natural compound. It was isolated from this little purple flower here, the rosy periwinkle. And so this rosy periwinkle, it was known as a folk remedy for many cultures for centuries for treating ailments such as diabetes. The plant is highly toxic. And again, I remind you, right? Oftentimes we think about, oh, that plant is poison. Or we think about, oh, don't put that in your body. It's poison. But what makes a poison a poison? Right? In this case, it's because it is a molecule or a compound that your body does not know how to deal with because it's not naturally found in your body. And as a result, by chance, the shape of this molecule interacts and interferes with other proteins in your body. Right? And the more important or the more disruptive that interaction is in terms of interrupting a very important pathway in the cell, the more poisonous this particular substance is. So in the 1950s, it was shown that this vincristine from the rosy periwinkle flower was very effective in leukemias in lab mice. And so then they began marketing this compound as Oncovin starting in the 60s. And so like the paclitaxel, it also targets the microtubules, but in this case, it targets and binds to the alpha tubulin monomer of the microtubule chain, and it prevents further polymerization of the microtubule. So the microtubule cannot grow, right? So therefore, the spindle fiber cannot even bind and reach the sister chromatids during metaphase. So this is the last slide for today. Next time, we'll talk about some more ideas with de novo drug discovery, because it's one thing to take existing compounds that were maybe discovered in nature and trying to improve them, right? Or compounds that were stumbled upon in the lab and trying to improve them. But what if you want to come up with something new, right? What if you love this idea that drugs are all about just three-dimensional shapes? And you know that there are certainly tons and tons of three-dimensional shapes that are yet to be discovered or synthesized, right? And maybe some of these shapes that you can come up with might be very important for disrupting certain pathways in cancer. So that's what we mean by de novo or rational drug discovery, right? De novo means from brand new, and rational means you actually know what you're doing. You are planning out a strategy to build something specifically for a particular target. So rational drug discovery means identification and invention of novel therapeutic compounds based on a biological understanding of what these target molecule pathways are that you're trying to get at. Right? So this is why I say this is very different than improving surgical approaches or radiation approaches. Right? You can only do this if you understand the biology of cancer, which is why at this point in our semester, hopefully all of you get kind of excited at this idea, because all the things that we've learned over the semester, all the underlying reasons and pathways for how cells transform and become cancer, this is where you can apply it. So when you're developing a brand new drug from scratch, de novo, you have to think about these four considerations. One is feasibility of the target. What is it that you're trying to target? What is the target space? Meaning if you're trying to design something that binds to another molecule, what is that three-dimensional space that you're trying to target? Is it a ligand binding site? Is it a catalytic site of an enzyme, or a site that just interacts with another downstream protein? Number two is specificity. How do you know that if this thing interferes with the two target molecules that you want it to, are you sure it's not going to also interfere with a number of other important pathways, right? Meaning the off-target effects might actually be even worse, more toxic to the body. You have to consider, number three, bioavailability. That means how will this compound that you come up with actually act inside the complicated physiological environment in vivo. Right? So different types of pharmacokinetics, pharmacodynamics. These are things we'll talk about next time. And then, of course, number four is efficacy in vivo. Right? It's one thing if you put this in some media on a dish of cells, right, in a plastic dish, and that does something, but how is it actually going to behave when it's in a living system? Is it going to target the tumor to the same extent that it targeted the tumor cells on the plastic dish?